And that would be, for example, taking someone with terrible contamination fears and handing them a gauze pad, which you have either really or told them you have put on a toilet seat. And there they are in the scanner holding that gauze pad, and presumably you've provoked terrible anxiety in their brain. Okay? And then the final sort of level of this type of study was during cognitive activation tasks. And this meant people thought, oh gee, these symptom provocation studies are sort of messy because you don't, when you provoke someone, you don't just provoke OCD symptoms, right? You provoke intense anxiety or general anxiety, but you also might provoke, you're also asking these patients to lie still in the scanner. So you've provoked all this anxiety, but then you've asked them to lie very still, so you've also asked them to inhibit what you've just provoked. So you see how it's actually sort of a little complicated to really figure out what you've done in the symptom provocation studies. So the next level of it was people said, okay, let's not do that. Let's go give them a cognitive task, which we know in healthy subjects recruits certain parts of the brain. We know that from other studies. And let's give that cognitive task, which doesn't necessarily provoke any OCD symptoms, and study patients who have OCD while they're doing those cognitive tasks to try to get at a cleaner sense of how the brain is functioning. Okay, so I'm going to just go through quickly. So these are the PET studies at rest. Most used FDG PET, which looks at metabolism. One used O15, which can look at blood flow. And between 87 and 95, it actually looked pretty good. I mean, this was really everyone got very excited about this because in brain imaging findings, these seemed pretty reliable in the sense that 5 out of 7 PET studies found elevated orbital frontal caudate metabolism or blood flow in OCD patients compared to controls. 5 out of 7 is pretty good. 2 out of 7 found increased caudate metabolism. 2 out of 7 found increased thalamic metabolism. Okay? What's the problem? Well, Saxena in 2001 looked at the largest OCD sample using the same FDG PET methodology. And he found increased thalamic metabolism, but actually only a trend for higher right caudate metabolism and nothing in the orbital frontal cortex. So the reason for this lack of replication in the largest OCD sample to date, we're not sure. Again, what people sort of go to is sample variability. That's sort of the buzzword now in the research literature. Oh, maybe he's attracted a different sample of OCD patients, and even the idea now is maybe OCD patients who have different types of symptoms have different types of functional brain abnormalities. So he's actually pursuing that by doing PET imaging studies just in hoarders, for example. So that's at rest. This is before and after treatment. And this actually created just such excitement in the field in the 90s well, not when the medication studies were done. When the medication studies were done, basically there were PET studies done before and after medication. And you can see that if you responded to medication, you had decreased metabolism in, interestingly, these same 3 brain areas. Orbital frontal cortex, caudate nucleus, cingulate cortex. This is what became so exciting was there were 2 PET studies that actually looked before and after cognitive behavioral therapy for OCD. And they found a decreased caudate metabolism. And that, when that hit the literature, that was like so thrilling for psychiatrists like myself who are interested in therapy, because it was really one of the first demonstrations that therapy can change your brain. Okay? That was Jeffrey Schwartz's work with Lou Baxter out at UCLA, and he's written about that work in a book called Brain Lock, which many of you may know about. Saxena in 2002, in that large sample where he didn't replicate the baseline findings, did actually replicate the before and after medication findings, in the sense that he also found decreased metabolism in the caudate, putamen, and thalamus. And then the symptom provocation studies, which I see someone wincing in the audience, so I imagine you experience this as this must be torture for the patients. And they found, again, different technologies were used, patients' symptoms were provoked, you image their brain, and there again, the same 3 regions keep popping up: orbital frontal cortex, caudate nucleus, anterior cingulate. And there are 3 different, very, very well done studies which showed activation in these areas when you provoke symptoms. Again, though, what are the problems? You activate more than just obsessions and compulsions when you do this. That, in fact, if you look carefully at those studies, they don't just activate those 3 regions. They activate other regions as well, and finally they're all samples, the sample sizes are quite small. And then cognitive activation studies. I'm going to just go quickly through this, but again, it's this, Scott Rausch up at Harvard has shown that giving OCD patients a task that in healthy controls usually recruits their basal ganglia circuits does not in patients with OCD. And they conclude that OCD patients may have a problem in those, again, in those basal ganglia circuits that healthy controls would use to do a particular cognitive task. So the conclusions are, from this body of functional brain imaging work, is that resting PET studies give variable results. The question is because of the variable state of the subject? But some do implicate the orbital frontal cortex and the caudate nucleus. And symptom provocation is associated with increasing activity, and response to treatment is associated with decreasing activity, again, in the orbital frontal cortex and the caudate nucleus. Again, I give you the caveat that what does increasing activity mean? Does it mean OCD symptoms themselves, or does it mean the effort to resist intrusive thoughts and urges? And there's a sort of fight in the literature around that because of brain imaging studies from patients with Tourette's disorder that I won't go through. But, and then again, there are other areas that people get interested in because of this brain imaging work, the anterior cingulate, the amygdala, the thalamus. So this has led to different models of OCD. And the dominant one is this striatal topography model of Baxter, Rausch, Saxena. Those are the sort of main names that have been writing about it and have been promulgating it. I just want to point, I'm going to sort of focus on this one, but I want to point out that there are some alternative models. Rosenberg has been doing MRS studies looking at glutamate in children out in the Midwest, and he's sort of thinking more about a slightly different model, and Carlson in Europe has a different model as well. But let me go through, instead of the words, again, the picture, which is the idea of it is again, from these functional neuroimaging studies and the structural brain imaging studies, people have focused on the orbital frontal cortex, the caudate nucleus, and the thalamus. We know that that's in a brain circuit in everybody's brain. Usually the orbital frontal cortex projects to this part of the basal ganglia, and that this part of the basal ganglia then can either inhibit the thalamus or not. And the model of Saxena and Rausch is that in OCD patients, and they're not, the model keeps sort of slightly shifting. Originally they focused on the problem being here. Because of Rosenberg's recent data of problems in the orbital frontal cortex and some other animal research in the orbital frontal cortex, Saxena and Rausch are now sort of saying, well, maybe the problem isn't here. Maybe the problem is actually in the orbital frontal cortex, or maybe the problem is some interaction between these two areas. But it doesn't, the details I don't think matter so much. The bottom line of it is the model is that this pathway isn't functioning appropriately, and as a result, the thalamus is not appropriately inhibiting stimuli that are going up into the cortex. Now really, Rosenberg's model, all he does is he focuses here instead of the whole circuit. And Carlson is sort of another variant on it. So really what I think the three of them are really sort of trying to figure out is what's the next level of this model to make it a little bit more specific. Which pathways are really problematic and which chemicals in which pathways are really problematic, and we don't know. So that leads me to neurochemistry. Okay, so again, the dominant chemical that people have sort of focused on in OCD is serotonin, and there's one main reason for that, because the only medications found to be effective in OCD are serotonin reuptake inhibitors. And so serotonin reuptake inhibitors act on something called the serotonin transporter, which is on a serotonin cell. So when a serotonin cell is about to release serotonin into the cleft, once it releases serotonin into that little space between it and its neighbor, it has on its cell what I call the little serotonin vacuum cleaner, which sort of sucks up the serotonin out of the cleft and, if you will, stops communication between those two cells. Okay? Serotonin reuptake inhibitors, the first action they have in the brain is to bind to that vacuum cleaner so that the serotonin cell does not remove the serotonin from the synaptic cleft, presumably keeping serotonin around in that space longer. Okay? Now that's not the only thing that these medications do. That's the first step. but in fact, then there are a lot of second and third and fourth steps. And if that was all these medications did, then people should see the response from their serotonin reuptake inhibitor like in the first week, because it happens in the first week. And as you all know, they don't. It takes 6 to 10 weeks for their medications to kick in. But anyway, because of the selective efficacy of serotonin reuptake inhibitors in OCD, unlike, let's say, depression, where there's a whole category of medications that can work in depression, because of the selective efficacy of serotonin reuptake inhibitors, that started the serotonin hypothesis of OCD. Huge caveat, just because a medication fixes something doesn't mean there's an underlying problem, that's the underlying problem in that condition. And you know, there are a lot of examples of that in the medical literature. But anyway, that's sort of the serotonin idea. Because some, and again, we'll get to this in the next talk, but because some OCD patients respond to dopamine agonists as augmentation treatments, dopamine also has been sort of thrown out there as possibly being a chemical important in OCD. Because of Rosenberg's recent work in MRS, he's really focused on glutamate. Glutamate's become sort of the hot chemical in OCD, and glutamate is one of the main excitatory neurotransmitters in the brain. It's all through the orbital frontal cortex, and the glutamate projection cells project to the basal ganglia. So for example, Rosenberg and Carlson's model really focus on maybe the real problem is in the orbital frontal cortex, and those glutamate neurons are like hyperactive, and they're mucking up the basal ganglia circuit so that the thalamus isn't appropriately inhibited. So glutamate's sort of on the map now. And Carlson actually even brings in acetylcholine, because her model is that acetylcholine is how glutamate gets messed up in the first place. There's very, very little neurochemical data in OCD. There are a handful of studies. I'm going to show you results from our group where we've done a PET study looking at the serotonin transporter. So again, I'm just going back to that, in case I've lost anyone, I'm just going back to that brain cell. And again, cell body of the brain cell, dendrites, which is where they communicate with their neighbors on this side of them, getting incoming signals from their neighbors. There's their cell body, there's their axonal process, that's their axonal terminal. And this is where their neighboring cell's going to be, so that they release chemicals from the axonal terminals into that little space. The next slide shows that. They release chemicals into that little white space, which is the space between this cell and their neighbor cell. And that's how their neighbor cell knows they're trying to communicate something with them. Now I'm going to go one level more complex, because you're going to need to understand that to understand the results I'm going to show you, which is they don't just squirt the stuff out there and the neighboring cell just sort of picks it up through their membrane. In fact, it's a whole complicated series or set of stuff. So this is now very, very blown up. This is the synaptic cleft, the space between the two cells. This is the cell that has the axon, which has the chemicals, and this is now one of those little chemical sacs dumping through their membrane into the space. So those little purple things are the chemicals coming out of this, what's called the presynaptic cell. This is the neighboring cell. It also has a membrane, and on its membrane it has multiple receptors for different chemicals. And it's those receptors that tell it specifically, oh, I just had some serotonin. But it even gets one more level more complicated than that, which is there are multiple serotonin receptors. And when one of those serotonin receptors feels serotonin, it does one thing inside the cell. But when a different serotonin receptor gets serotonin bound to it, it does something else. So it rapidly, the whole neurochemistry of the brain gets very complicated very quickly. Okay? What we wanted to do, because again, this right here is trying to show you that little vacuum cleaner on the presynaptic cell, picking up serotonin out of the cleft and taking it back into the presynaptic cell, and if you will, saying, we're not going to talk anymore. Okay, I'm going to take away the serotonin I gave out to you. I'm done the conversation, I'm hanging up. And if you will, the serotonin transporter lets that presynaptic cell say, conversation over. Serotonin reuptake inhibitor, or it's called also the serotonin transporter, that's where the serotonin reuptake inhibitors first bind in the brain. So as we're not brain imaging specialists. We are specialists in the anxiety disorders clinic. We do diagnosis and treatment of different anxiety disorders. And yet I believe, and we believe very strongly, that if we understood better the brain basis of these disorders, we could actually long-term, ideally, produce much better treatments than we have now. And so we have collaborated with a brain imaging group at our institute, Mark LaRuelle's group, who's one of the leading experts in a very particular type of brain imaging technology, which is what's called neuroreceptor PET imaging. So this is a way of, his group has made a whole bunch of compounds that actually target specific neurochemical either receptors or transporters in the brain. And he's developed a methodology for how you can actually image that in a living person's brain. Now this is actually very exciting because up until SPECS started to do it, and now PET's doing it, and now MRS is doing it. So it's a whole new wave of functional neuroimaging, which is actually looking at neurochemical imaging. And it's very exciting to us because up until this point, there was no way you could image the chemistry of a living person's brain other than after they had died. And as I told you, there are no post-mortem studies of OCD patients. So we collaborated with him on the study that I'm going to tell you about, which was to look at the serotonin transporter in OCD patients versus healthy controls. So the aim was, the serotonin transporter is known as SERT, okay? So the aim was to study the availability of SERT in the brains of untreated OCD subjects and match them to healthy controls using PET and this particular ligand that Mark LaRuelle's group developed, which is called the McNeil ligand. The reason why you have to carefully match patients is, as you imagine, the serotonin system, which is in all our brains, changes if you're male or female, changes with age. So you have to, we don't know about ethnicity, but you could even imagine is different between ethnicities. And so our goal was to recruit a group of people with OCD and match them to a group of people who didn't have OCD, run them through the exact same brain imaging procedure, and at the end of it ask, is there differences in the availability of the serotonin transporter in particular brain regions between if you have OCD versus if you don't. Is that clear? Yes? Okay. So our aim was, as I said here, our hypothesis, is OCD is associated with decreased SERT availability in the caudate nucleus. And there's a variety of reasons why we actually came up with this hypothesis. I won't go through it because I'll give you the answer now. We found nothing. So I'm not going to belabor our hypothesis. And that decreased SERT availability is associated with severity of OCD symptoms. So let me explain first. Yes, this is just to remind you again, what we're going to look at is the availability of this protein, the serotonin transporter, in different parts of the brain. For technical reasons, this compound that Mark LaRuelle developed to be able to visualize this particular protein can't look anywhere in the cortex. So we couldn't look in the frontal cortex, which is obviously very disappointing to us since the orbital frontal cortex is thought to be important in OCD. What we could look at was the caudate, the thalamus, and other parts of the subcortical nuclei. Okay, I'm actually going to step back for a second. I think I'm going to just describe this as opposed to put that up. How do you do a study like this? So basically, after recruiting people, there are two things people go through. One is a PET scan. And what a PET scan is a PET scan is for the neurochemical data. And Mark LaRuelle's group, as I said, has this compound called McNeil that binds reversibly to the serotonin transporter. The PET camera can't see that compound. So that compound is tagged with a radioactive particle called carbon-11. That compound, plus the carbon-11 particle, which is combined with it, gets injected into the vein of a subject. The bloodstream takes that to the brain. That compound binds to the serotonin transporter where it is in the brain, unbinds to the serotonin transporter because it was reversible in the brain. The body then metabolizes that compound and excretes it from the body. And the carbon-11 particle has a half-life, the radioactivity has a half-life of about 20 minutes. So within about 3 hours, the radioactivity from the carbon-11 particle is all gone. During that study, it's not enough to just put a subject in the camera, take a picture, and everybody goes home. Because people get slightly different amounts of the chemical to start with, and people's bodies metabolize the chemical at different rates. And so during the time that they're in so the bottom line of it is we're trying to find a particular point in all subjects, which is where the amount of the chemical entering the brain exactly equals the amount of the chemical leaving the brain, which is called an equilibrium point. And depending on the chemical that you use or the ligand you use, you have to be in the PET camera for different amounts of time. And while you're in the PET camera is like collecting pictures of the brain. But that's not even enough. Because the compound is degraded by the body as soon as you inject it, and the radioactivity is decaying as soon as you inject it, multiple small samples of blood need to be taken from the subject over the course of the PET scan. And that blood is then analyzed for how much of the parent compound is left and how much radioactivity is left. And then through quantification, basically you come up with, through all of this, through the pictures from the PET scan and the blood data, you come up with how much of the serotonin transporter is specifically in different parts of the brain. Okay? As you can hear from what I'm saying is there are risks to the subjects for this. And so this is research that I really view as sort of state-of-the-art scientific research where our subjects are our participants. We couldn't do research like this if subjects didn't agree to it. And I feel very strongly that people don't go into this unless they really understand it, because there are risks to them in terms of exposure to radiation and exposure to an arterial line. Obviously, we don't get, the FDA makes guidelines for how much radiation a subject can be exposed to. I'm not allowed to just give anybody any old amount. It's very tightly regulated how much radiation you can receive as a research subject. It's regulated how much radiation you can get if you're a worker around radiation. What's not regulated is medical treatment. Like if you have prostate cancer and you go for radiation treatment, that's not regulated because it's assumed that the cost of radiation exposure is worth the benefit. But the federal government gives very strict guidelines about what we're allowed to do with research, and of course our studies are below those guidelines. But the reality of it is there's still risks to the subjects. We do everything we can to minimize them, but they're risks. So what I'm going to show you is, I feel very grateful to the subjects, both healthy controls and OCD patients who agreed to participate in this work, because without their taking on this risk, we couldn't move science forward. Okay. So this is just describing the PET procedures and the imaging processing. This is showing how the quantification, and the bottom line of it is don't worry about the details of it, but the bottom line of it is we end up with two outcome measures. One's called binding potential, one's called V3 double prime. The differences between them, again, I don't think is important for you to understand. Just to say that in this type of literature, there's a big debate about which one is better, and we just did both. And again, there are different ways, both of those outcome measures are different ways of measuring the availability of the serotonin transporter, that protein on the presynaptic cell. Now this is to remind me that all patients not only got a PET scan, which is a study that happens on a single day. The patient's in the camera for about 2 hours, but with setup and stuff like that, it's usually sort of a 5 or 6 hour procedure. They also come in and get an MRI scan. And the reason why they get an MRI scan is that the PET scan gives us the chemical data, but it has very, very poor structural resolution. So you get fuzzy green pictures from the PET scan. And we're not interested in just the serotonin transporter anywhere. We're interested in the serotonin transporter in particular parts of the brain. And so all subjects also get an MRI scan. And an MRI scan is like an atlas of your brain. And by getting an MRI scan, we then overlay the PET pictures on top of the MRI scan, and we can draw on the MRI scan particular parts of the brain, like the caudate, the putamen, the ventral striatum, the midbrain, the thalamus. We don't have to guess where they are. We can actually outline them on an individual person's brain and then overlay their PET data. So we can actually ask about the availability of the serotonin transporter in these particular brain areas. So this is actually a picture. This is showing you a slice like this through the brain with ventricles, and this is actually showing you a part of the basal ganglia. It's showing you the dorsal caudate, dorsal putamen, ventral striatum, but it's really just to illustrate how we take an MRI slice like that and then literally draw the different parts of the brain on it, and then add up how much dorsal caudate volume this subject has versus that subject has. The other thing this slide's to remind me of is, again, to remind you that these are the brain regions we can look at. And again, we could not look at the orbital frontal cortex for technical reasons from this ligand, but we could look at the midbrain thalamus, different striatal regions, or the basal ganglia, different limbic regions. The amygdala is of interest in a lot of other anxiety disorders. It's one of the parts of the brain very important in something called fear conditioning for people who know that literature. Okay, so we recruited people with OCD, and we also recruited control subjects. And these are really fussy really fussy studies, which is we can only take a certain age level. Obviously, they all have to have OCD. They can't have any medical or neurological problem. Obviously, they can't be pregnant. They can't be on medications because we want to study the chemistry of their brain. If they're on medications, presumably their medications have altered the chemistry of their brain, and we don't want to study their altered brain. We want to study, if you will, their unaltered brain. We didn't want comorbidity because we're trying to focus on OCD. There are a lot of hypotheses about serotonin and other anxiety disorders. There are hypotheses about serotonin and major depression. So anyway, and then obviously control subjects, we needed everything except the OCD. And this was the sample. We ended up with 11 OCD subjects, but this is just showing you sort of different profiles, different scales. I won't go through the details of that in the interest of time. This is just showing you how we sort of do the scan parameters and showing you curves of, this is injection of the compound into the vein and how the metabolite and the radioactivity decline with time. This is the uptake, this is showing you just for the cerebellum. This is the uptake of the chemical into the brain, into the cerebellum, and then starting to wash out. And again, what we're looking for is this equilibrium point, which is, my hand's wavering, so I can't quite point it out. But as you see, is where the amount going in, it's exactly equal to the amount going out, because that's a point we can find in every person identically, and that's where we compare our data points. Okay, so the first thing we looked at was what I call region of interest volumes, which is all those different brain areas: midbrain, thalamus, dorsal caudate, dorsal putamen, ventral striatum, amygdala, hippocampus, cingulate cortex. And we looked, oh, I'm sorry, gosh, what's blue and what's white isn't on the scale. Well, I don't remember whether, I think OCD is blue and healthy controls are white, but the bottom line of it is there was no difference. Okay. And that's good. I was going to say, let me go back for a second. That's good because if you had huge volume differences, then you have like a mess of how do you control for that. So we didn't find volume differences. Then we looked at one of our outcome measures, which was binding potential. We found no difference there. Then this is looking specifically at the caudate nucleus because that was our particular interest. And this is showing you instead of average, I'm sorry, I didn't explain this slide very well, did I? This is the mean. You take all of those 10 controls, you take all of those OCD patients, and you average their value, and this bar is the standard deviation. Okay? So these are mean values for the group, and that's what we're comparing there. Mean values for the two groups over the different brain regions. This is now giving you the individual values for each of the individual subjects. Okay, so these are the control subjects, these are the OCD subjects. This is looking in one brain region, the dorsal caudate. This is looking at one outcome variable, the binding potential. And what you're seeing is that, yes, there's more spread in the OCD group, but you're also seeing that they're really very overlapping. And the bars there are the mean bars. And this was true for all the brain regions. So that's binding potential. We found no difference in V3 double prime. Again, these are the different brain regions. These are the mean values and standard deviations for the OCD group in blue, the control group in white, and this is the outcome measures, V3 double prime. Found no difference there. Okay, so unfortunately, when you get negative results in science, even though this was sort of the good thing of this was surprising, so that was interesting, right? Although it would be better for me personally if there had been a result because I could have written a grant and could have continued doing research. So there's on the other hand, from a science point of view, this was interesting, right? Meaning whether you got a positive answer or a negative answer, this was interesting because I feel like it moves the field forward a notch, okay? However, when you get negative results, then the onus is on you to really demonstrate that they were really negative, right? They weren't negative for artificial reasons, but they were really negative because you looked under every doormat, every different way you could look at it, and they were still negative. So the next two things I'm showing you is basically just our trying to do that. So this isn't for you to read, but this is to remind you the other way you can look at this sort of brain imaging data is what's called voxel-wise analysis. So one way is what's called region of interest, which is I say, I'm interested in the caudate nucleus because that's what the brain imaging studies have shown, that it's an area of importance, the functional neuroimaging studies. I want to look particularly at the caudate nucleus. But what happens if the abnormality in